make omega-3 tablets and also to sell for fish food for aquaculture. So I'm grateful for that because I feel like this is such an important part of the food web here on the West Coast, and I'm glad they had some forethought to really look at the ecosystem as a whole and ban the harvest of krill.
And sometimes it takes these policies to create these positive impacts or create the change needed to support the health of an ecosystem. So there's an interesting relationship between science, research, and policy often.
Okay, so my relief is here. Yeah. Thanks a lot, guys. And,
thanks everyone for tuning in. We're the 4 to 8 watch, and many people within our team are getting relieved by the next watch, our 8 to 12 watch team. It was such a pleasure, and we so enjoyed your questions and your comments, so keep them coming. And again, just to kind of remind you, we're in the Greater Farallones National Marine Sanctuary. We're on a 22-hour dive. We deployed the ROVs yesterday afternoon at 4 p.m. So we're well into the dive at around, 16 hours in. So stay tuned to continue seeing krill, rockfish, and hopefully some great diversity on rock substrate.
I have a fun rockfish fact to share from Tom Leidig from National Marine Fisheries Service before we leave. Somebody was asking about rockfish behavior, and Tom says that chili pepper rockfish, which we've been seeing quite a bit of, they have two different patterns that they can change through these special cells that they have in their skin called chromatophores. And they have these two patterns. They have bars when they're on the seafloor, and then change that to have a solid color when they're in the mid-water. So I thought, but we were just seeing the lighting picking up different patterns, but they're actually changing those patterns on their skin through these special cells that they have. Which I think they do through like expanding and contracting these cells, but I'm not sure. So that's pretty neat.
Thanks again, all. I'm going to hand this over to Brandy Jones, our Science Communication Fellow, and I'll talk to you all later.
Another fun fact about krill is that they can change their body size if there's a lack of phytoplankton. They'll just shrink, which conceivably should work for humans too, right? If we don't eat, we should shrink a little bit. It's a little harder for mammals, but they just adapt, get smaller. So that's one of the things that we look at when we're looking at the prey samples in the access program we do at Sea Monitoring. And our partner, Point Blue Conservation Science, they do krill toes, and they count the size. They count krill to estimate abundance, but also look at the size of the krill, which gives an idea of the health of them eating. And if they stay small, that means there's not as much food for upper levels of the food web. And krill also have photophores. They can bioluminesce.
There, he's stuck over there. Sorry. Let me know if you want me to hold up here.
Good morning, team.
Good morning. Looks pretty clear as far as terrain goes. You in a better spot now? I know, one of these days. Hmm? Hello, science.
They don't want to talk to you.
Their headsets aren't on.
Oh,
Jamie, that's why we don't fly backwards. What was that about flying backwards?
That's why we don't fly backwards.
Why don't we fly backwards?
You know why?
Oh, yeah, I do know why. Okay. Roger that.
Do you fly backwards in the southern hemisphere?
We had to. That was the only way we could go.
Yeah.
Yeah.
Coriolis.
Yeah, Coriolis.
I was gonna say we turn in opposite directions.
Yeah.
The foam pack's on bottom. It's all weird.
Yeah.
Oh my gosh.
So we're heading along this straight track here.
Is that a slope map?
That is a slope map. So it's pretty not slopey until that slope right there.
That was really descriptive, Rennie. Thank you.
Well, that's a technical navigator term.
Yeah.
Yep.
Like the scrape and slurp, except that wasn't a navigator term.
That was
an ROV term.
Well, scrape and slurp. I said, oh, we're going to scrape and slurp, and
It was a technical term.
it was a technical.
Yeah.
Exactly.
We do use
Yeah.
that.
We like to keep our technical terms
Technical.
more layman's terms.
Yeah.
You know?
Jargon for everyone.
Yeah, because
Yeah.
like we could call a scrape and slurp something like
What's he gonna
pulling an
come up with?
Alfonso.
Yeah. Well, we do say William script.
Hey, Rennie, do you have
Yes.
on your screen somewhere the time end of that last transect?
The end?
End, yeah.
I think the last transect. I don't.
Yeah, the very last transect. It just didn't get recorded back here.
Yeah.
Don't worry, we can get it.
Yeah, I'm not sure.
Okay.
Just trying to look at the last time they went straight, and it looks like
How
they
did
were,
numbers look?
they were driving.
I think they ended like right during the shift change.
It could have been just they might just be going by the targets rather than dropping their own.
Okay.
So I don't know. Then if that's the case, I don't have the timestamp.
Okay.
Welcome, world. This is Brandy Jones, the Science Communication Fellow from Houston, Texas.
Video
Thank
fish.
you for tuning in.
Eat a krill. You know you want to. Boy, rockfish are hard to train.
Even worse when they can hear you.
We convinced somebody one time that there was a hydrophone on the vehicle. So I'm full wide. Yeah, we were zoomed in on a vent, and the bacterial mat had grown a tube around this one, I guess, vent orifice, and so you'd got this, it would fold over, and then the bubble would, you know, kind of like bloop out of it. So I think Brennan started making noises, lined up exactly with the air bubble coming out. Well, there's not as much krill as there was. What is that? That is square.
Kind of looks like a... No, it's
Video
too long to
pushing.
be a brick.
California long brick.
It's a piece of
Piece
wood.
of wood.
Piece of wood.
Yeah, like a 4x4.
It's been
All right,
down there for a
full
while.
wide. Yeah, it's pretty buried.
It's pressure treated.
Oh, Rennie, you're losing your touch. Jan's killing it back there.
It's the morning.
I learned from the master.
From George.
Man, that's now Paul and
Well
Jan are
done,
both on in
Paul.
the morning. That's good. I'll take the night watch. You guys get to have this one.
Jan, Drew says hi and that you're krilling it.
Drew, I used that last night, and they all thought it was so funny.
The previous watch was just saying that as I came in, too.
Yeah. Loses its appeal after the
Yeah, you
10th
find out
time.
another, well, I was going to say, you find out another watch is doing it. It's just like, wow,
Wow.
Yeah.
it's blasé at that point.
Well, Drew and I,
little
and Danny, we work on a
dark.
separate project where
Video
we look
pushing?
at birds, mammals, and krill.
So the
And
merchant,
we actually have
an enemy.
T-shirts that say, "We're krilling it."
I love it.
Is that just an anemone that's buried?
Yep.
Yeah.
It's a tube anemone that's in its tube.
Yeah.
What kind of fish is that?
One that's not eating krill.
Well, we have Tom Leidig on Science Chat, so we can swing. There we go. Is it a thorny head that is an odd color? Icelinus sculpin.
Oh, I thought it looked sculpin-like. I wanted to say blob sculpin, but I was like, it's not a blob
Not a
sculpin.
blob.
No, we're not deep enough for a blob sculpin. Gonna look that up, Tom.
Wow, it's really in there, huh?
Cool. Yeah, I think they work to keep sediment out and build the tube up as the sediment rises. Full wide.
There it is, Gary, on the screen. Yeah, it's much darker in the photos. So, Brandy, one of the things we do is we share photos, especially photos of rockfish from the side, full body, with other scientists, people much more knowledgeable about rockfish, and say, here you go, here's a nice photo of a rockfish. Can you use it for your ID guide?
Awesome.
Now, is that a cow cod? No.
I don't think so. Is it a green spot? I'm looking at Tom's feed.
Video pushing.
No, it's not a green spot. Oh, here we go. You can zoom in on as many as you can so we can count the fin rays. Oh, I think he was talking about the sculpin there. Then we can tell what species. No, that is a green spotted. Ah, yes.
Oh, you were right. You just got to have more faith in yourself. Full wide.
Just a shout out to Tom. Thank you so much for being on a line. And I was the rockfish expert after everybody got off Science Chat. And eventually Erica Burton got on and saved me
Yep.
last night.
They were no longer all just rockfish.
It's a big one.
Good morning,
Oh,
Miss
he's
Cole's
doing it.
class.
Oh, it's got a little cave.
I feel like this is one of those you can't see me now, but you know it's just because your eyes are covered. All right, we're gonna go back out in front in hopes that, you know.
So that big one in the cave was a cow cod, confirmed by Tom.
Someone
Not a
in the
rockfish.
live feed would like to know, does this large number of krill affect the turbines on Herc?
No. The thrusters and the propellers on Herc are aluminum, cast aluminum, and they are operating at 3,000 psi. That's the hydraulic pressure for spinning them. So they are more likely to cut through something. It's one of the reasons that we're pretty careful about not having fish fly into the vehicle from the front. One of the things that helps with that is just Hercules moving through the water creates a pretty decent pressure wave, and so it kind of pushes the fish away anyway, or it scares them off. But there are times where like a fish will swim past the main HD and we'll kind of back up a little bit, try to blow the fish out the front of the vehicle.
We got some terrain coming up. Just make sure we're out in front.
We're at video pushing. It's a little new to Brink.
Oh, yeah.
All right, Aaron, let's come full wide. I don't hear clamoring in the back, so. Or like rapid page turning.
We already have that one.
Mm.
Yeah, it's in the container. Yeah. Oh, I don't know if it's in one piece. It should be. It was pretty gentle.
For a while, we were having issue after issue with the suction pumps, or the pumps for the slurp, and we just kept replacing one after another and rebuilding them, and then this one's been on for like 2 years without any kind of issue.
Dang.
Yeah.
Was there a difference in the design, or?
Same pump. Oh, that's a Dark Sea Star. Sea Star has bands on it. I'm still out in front, ready.
Mm-hmm.
I don't see a lot on the sonars.
Yeah, I think it's gradual. I don't think it's too bad.
Let's see, 169. So, Aaron, what do you want to see on this watch?
Still a jellyfish.
We saw some jellyfish last
did?
watch.
Yeah.
You weren't satisfied with those?
No, those were unsatisfactory jellyfish.
You're hurting their feelings.
I'm just glad she didn't say krill.
No, I'm over krill.
I'm happy because this ecosystem's quite abundant in krill, but I'm unhappy because they get in the way of the camera. Another octopus would be nice too.
All right, Aaron, let's push in. Yep, always on the nose. All right, let's back out and frame it. And we're off.
What are you hoping to see, Jamie?
I feel flexible. I don't have any particular needs or desires.
No agenda.
I'm open to options.
Yeah, we've come up 3 meters. So,
Yep.
Gradual slope.
Someone in our live feed would like to know what's the team's favorite encounter with a fish.
For me, it would have to be a flamboyant squidworm.
Oh my.
Flamboyant squidworm.
Video push in.
Not a fish. Illegal move.
Hey, Rinny, I'm going to come over and point a couple of new waypoints.
Sure thing.
We've got some interesting feature just south of us.
Yep.
Okay. Oh, look at that.
All right, full wide.
Wow.
Hey, you're not supposed to look at it.
We have someone on the live feed wanting to know what's the deepest you can dive to, so they're trying to settle an argument.
I didn't hear the question.
What's the deepest
Yeah.
that we can dive to?
The deepest that we can dive to is 4,000 meters. That's primarily limited by the flotation on Herc, which is only rated to 4,000 meters. Argus on its own could hit 6,000 if we just removed some of the instruments. But we just got finished a rebuild on Little Herc and built Atalanta. And those are both 6,000 meter rated. They were tested on the previous cruise.
Okay, so we're gonna head, just explore this feature of
heading south.
Ish, yeah.
And then southwest. I pay attention sometimes.
Let's go, we'll go 175 for a while. Just kidding, we will not. Go 165.
That looks like fluid.
Bridge now. Let's take it one step, 40 meters, bearing 165. Thank you.
There you go, bacterial mat.
I decided what I would like to see on this watch. I want to see a lot of really tiny critters, like that baby sunstar up there. Very cute.
I call it a step. Let's think about if we want that to be
is the group.
Rathbanaster. Or Rathbanister, as I like to say.
For what? And 175?
165.
65.
It's only about 140 meters away. I don't think we should track a line there. I think it's gonna be more trouble than it's worth. I'll call in a few steps and.
All right. All right, Paul, I'm gonna come over so you can come over to 165.
Can we zoom on that little anemone?
Aaron, push in. Oh gosh. Forgot how fast it moves at full zoom. Are you talking about the little one next to it, or?
No, the big one, and you've got some good captures of it. I'm happy. Yeah, it's lovely.
It is. Colors
All
are
right.
beautiful.
Jamie, do you have one with the lasers?
Sure.
Lasers.
Lovely. Thank you so much.
Full wide. What you doing over there, Rennie? Aaron, let's push in. Crab and sponge.
Where's the crab?
Oh, there's a crab.
Oh,
Oh,
it's
nice.
disguised. I didn't see it through the camouflage.
Let's add one more step.
So
Thank
it's
you.
a little crab at the top. All right, full wide.
Jamie, did they get a similar sample last night? I know they got a sponge.
Let's see how they described their sponge.
Basket star.
They got a light yellow sponge. We got that globular finger sponge. Coral, palm frond sponge, nudibranch.
Yeah, we did all those.
Cup coral.
Oh, they did a cup coral.
And then some brachiopods. So no, we don't have any sponges like that collected this dive.
Is that a we want to go back for it, or we want to keep an eye out for one?
I haven't seen it with those big, what are they called? Oscules? Yeah. Yeah, we could.
Do you want to? I'll have to stop the ship and go back.
Yeah. Oh, if you have to stop the ship, no.
There's a few up here. If we're interested,
Yeah.
we could stop right now.
Well, are those the same ones? I'm
Those
sorry, I
look
was
different.
typing.
Yeah, they're a bit different. They're
The-
flatter.
Yeah, and I know we got some samples of those.
Okay.
Either yesterday or the day before.
Aaron, zoom in.
But these
That's
sponges
looking
are
pretty similar.
hard. Yeah. But it doesn't have that big
Yeah.
oscule? That big hole.
I think it looks pretty similar.
Yeah, I think that's the same. Full wide. Still gonna push back out.
Yeah.
Favorite fish interaction? Was that one of the questions earlier?
Yeah.
Mm-hmm.
I think the
So let me
I'm
know
a-
if you
I'm
want
a
to stop
fan
for
of
any
the
of these, Jen.
predatory events, or predation events. So.
Oh yeah, that one last night was kind of.
Yeah, we had the lingcod with the rockfish in its mouth,
Yeah.
and then the sea star on the rockfish.
Yep. Yeah, if I had to choose which rockfish to be, it would be the lingcod rockfish, I think.
Yeah.
I don't know, the sea star rockfish still had a chance.
You know, that was kind of interesting. The lingcod had the majority of the rockfish in its mouth, and then we saw it
So
a
Jen, we're
later
held here.
point,
Do you want to
like,
keep going?
the fish had worked its way out a little
Yes.
bit, you know, so.
Bridge now. Add one more step. Thank you.
Some encrusting sponges, a crinoid.
I'm going to do a reset on the fly.
All right, well, I can just hold here for a little bit.
That's all right.
Video, let's push in.
It's that good. We're only at 100 some meters.
I'm really glad you decided to zoom on that because I wanted to see it too.
I had a feeling.
Lovely sea star.
All right. What else we got?
So, Jamie, did you notice that we have a new series of waypoints, and we're going to hit this feature and it's going to go outside of the management zone and then back into it.
Okay, so we're crossing from a closed zone into
an open zone.
Okay.
I don't know. Do you have it on your?
That sponge
Is that
looks pretty
that
much the same.
sponge
Yeah,
that
that
you
one looks
liked?
that one looks similar. Jan, if you want.
Is it?
Same shape.
I think it's just a smaller version. Video full wide.
There's some internal debating going on back here about the sponge.
Yeah.
Okay.
Yeah. Can you go in close, Aaron?
All right, video, let's push in.
In a minor retrograde. Oh, we have the leash.
I don't think it's the same one. Yeah.
Okay.
Thank you.
Okay, full wide.
Jamie, I don't know if you've seen earlier that little puffy sea stars. Someone on the live feed would like to know what type of sea star that is.
Like the one that's in view right now, that little yellowish tan one?
No, I think it was white.
The little tiny white one that we zoomed in on a minute ago? That's the extent of the description I have for that one, unfortunately. The little yellowish one we
can
just
head down a little bit
saw
on Delta.
is commonly called a cushion star or a bat star, but the little cute fat white one that we saw, I'm not sure what that was.
I thought it was another type of cushion star.
Oh yeah? Okay.
Yeah. But I left my sea star guide at home.
Oh.
It's okay. While we wait on the ship move, video, let's push in. Here you go, Jamie, tiny sea star.
You're fulfilling my every wish.
And if I'm correct.
Oh, that one's extra cute. Something about those when they're small.
The coloration's different.
Yeah.
All young animals are cute.
They are. It's just a fact. Oh, there's a really tiny sea star. Oh, and the little, is that a hermit crab or a snail moving?
Looks like a
Hermit crab.
It's fun to zoom in really close sometimes. There's so much more than you realize when you're just zooming over the seafloor. There's a lot going on.
Yep, I figured we have a
That's
moment.
why we need experts like Miss Erin.
There's a little tiny
hooking us up.
red shrimp. Very tiny brittle star. Some kind of little
There's
snail.
a little hermit crab in the
hermit crab.
top.
Oh yeah, that one's definitely a hermit crab. I wonder if the middle one is also a hermit crab.
Good morning, Don. Thanks
Numerous
for tuning
brachiopods. Lots of little tiny encrusting sponges.
Oh, and the vehicle.
Yeah. Very rare Hercules in the wild. It's natural habitat.
think that's one of those branching sponges. What else have we got?
Someone in the audience would like to know, will there be a new ROV in the next season?
Oh, there's a tiny coral. Looks
Yeah.
like the one we collected before.
That little red
Very
one.
tiny.
Yeah, it's super tiny. Aaron, let's come wide and get the lasers in the shot. Yeah.
Are you sure? Or isn't that one of those little squiggly things that we
No,
thought?
it doesn't have the same continuous spirally thing.
You mean the little worms, Rennie? Or are you
Yeah.
talking about something else? No.
I didn't see
All right.
the polyps on this.
I think I
Full
saw
zoom.
retracted
Let's
polyps.
do full zoom again. Let's.
Yeah, I think that's the squiggly worm.
Really?
I don't know. It's so tiny. All right,
Yeah.
full wide. We're gonna.
That's not polyps.
A coral wannabe.
Mm-hmm.
Coming up.
Aaron, push in on sponge. Is that the same one? Oop.
That looks like it has pretty big ossicles.
Yeah.
Yeah, that does look similar.
That looks, yeah. Looks,
Do we want it?
yeah. A little piece.
Sure.
Like that little lip that's sticking out, if
Bridge
that's
now.
easiest.
Hold position.
Video full wide.
You should be fine there.
Yeah.
Marcus might have to change heading, but I tip the cam down. But aside from that,
Hey, BJ.
All right, Aaron, let's push in again. Hold that. All right, those are lasers on it.
Got it?
So yeah, you could probably swing your heading around and get me back in frame. All right, let's come full wide. I'm gonna try and
the other way.
Yeah.
set down on this ledge.
We have some third graders wanting to know, have we seen any clams?
I don't think we've seen clams, maybe some clamshell fragments. Most of the little two-shelled organisms we're seeing are brachiopods, which is actually a different group from bivalves like clams and mussels or scallops.
Dan, you were talking about the upper lip or the lower lip?
Whichever's easiest.
Okay.
And not the crinoid preferably, unless you want a crinoid.
No.
Okay.
But I do like that upper lip. Those ossicles do look very different.
Sure.
Someone in the audience really wants to know, will there be a new ROV in the next season, since you were conducting tests with a deeper diving platform?
You know, I don't know. I'm assuming. All right, video, let's push in. Hold that. Might actually be easier to go for that top bit. Jamie, where are we going to put this? Do we know?
Depending on how floaty it is, we have a forward bio box available, or otherwise I would probably put it in the aftmost starboard small compartment.
Is that enough, Jan? Do you want more?
A little more. Just back a little bit further. Yeah, that should do.
You got yourself into a pickle.
All right, video full live. Let go.
Oh, got the whole thing. Nice.
All right, let's put that in the forward compartment. So we're gonna
Okay.
put it in Omega.
Oh wow! So
Nice
that
sample.
Just sheared off right at the base.
Yeah.
Oh.
Jamie, do you want a close look for associates?
I think we're gonna be able to put
if
in
you
its
go
own
to
container, so
hotel
it's okay.
first,
Yeah.
Okay.
and lights. Oh right, we haven't rebooted the GUI. That was on the to-do list, but it didn't get
Roman
done.
He's got
Hotel.
Roman
Yeah,
Hotel.
Roman Hotel.
I'm on it.
Miss Cole, we've seen some small sharks, but I think the water's a little too
Ready to
deep
open
for
the tool tray?
turtles.
Or
Just a moment. I don't really see a lot of associates on it.
Yeah.
Okay, now we're ready for tool tray.
okay.
All right, and Omega.
Yes, please. Nav, that's 040.
040.
All right.
Tool.
Yep. Tool tray in.
We have some students
and
wanting to
before we-
know if there are any squid or
Before we leave?
seahorses in the
Yeah.
area.
If
Zoom
we could get the
back in.
lasers on that, what's left of the sponge,
Sure.
just to make certain that we've got good measurements.
Yeah, we got a visual with the lasers on it before.
Okay.
Video, let's push in some.
Yeah. And actually, it'd be interesting to get some screen grabs of just the
broken bit?
yeah, the broken bit.
Sure, all the way in.
Jamie, can you let some students know what do you do with the organisms you collect?
Ask me again after we finish looking at this sponge.
Got it.
Thank you.
She's furiously typing away.
And she's taking photographs.
I'm actually all caught up, but I want to make sure I get the grabs we want for this sponge.
All right, everybody happy?
I'm happy.
Could you zoom out just a touch for
Sure.
1 second? That's good. I'm happy.
All right, full wide.
Okay, ready to continue on?
Yes.
I'm just gonna step a little bit south and then we'll go along our other wall. Bridge nav.
And we can turn off Roman Hotel.
One step, 25 meters bearing 163. Yes, thank you.
All right, so in terms of samples,
Precise.
So once the ROV is put back on deck at the end of the dive, we take all the samples off the vehicles, put them in little containers with water,
All right, I'm gonna
and
come
put
around
them
so
in
you
the
can bring your heading
fridge,
back
and
around.
then one at a time, we take them out. We leave them in the fridge initially because they came from cold water, and so the fridge is more reminiscent of that temperature than room temperature.
To remember the good times.
Process as quickly as possible.
And then we take them out one at a time. We photograph each sample with a ruler so that we have a good sense of its size and a relative scale. And then we take off any associate organisms. So for example, if we picked up a coral that had several brittle stars on it, the brittle stars are the associates. And we take those off, photograph them separately, and then
Should I come down a little bit?
Yeah, you're good to come down.
we take the different pieces of samples and we preserve them in little bags or jars with ethanol, which will keep the DNA intact, and then we send them back to labs on shore that can then do studies of their shape or morphology, as well as studies of their genetics, and look at all sorts of different pieces of information about the various organisms. So that's for the biology, and then for the rocks, we tend to take off any organisms, preserve those in the same way as what I just mentioned, and then dry the rocks, and they can go to geological labs that can study their composition.
Are we still working with Dean?
Thanks, Jamie.
We send rocks to the Graduate School of Oceanography at the University of Rhode Island. I don't know the direct contact. Dean doesn't.
No, Dean was is near enough to San Pedro that he's visited the ship. He sailed once, but he was taking rocks that still had all of their associates.
Can we look over to the right?
Over to the right.
That feature, is it a pipe? Is it rock?
I think it's rock. And so then
Guy's gone.
you didn't have to pick off the associates.
Or that
Just went into a
those
bag.
layers.
Gotcha. Yeah, that would probably be a cruise specific
okay.
choice.
What are some common types of fish do we see in this area? I noticed a lot of rockfish. Any other types of fish that we see in the area?
Sculpins.
Sculpin.
Yeah. Sculpins in the shallower area. Lingcod. Hake. A lot of hake around.
More rockfish.
And more rock, yeah.
Chimera.